Everyone, and welcome to the Hooked on Ocean Acidification mini-series. I'm Kathleen Davis, Project Coordinator at Maracus, and I will be your host for this evening. Tonight is the third night of our four-part mini-series. We're thrilled to see over 150 people are registered for this evening. And while this workshop was geared towards the recreational fishing community, we've attracted a broad group ranging from social scientists to fisheries managers to oyster gardeners, educators, and watershed ambassadors. We are looking forward to a very rich discussion this evening. Hooked on Ocean Acidification is sponsored by the Mid-Atlantic Coastal Acidification Network, MACAN, and the Mid-Atlantic Regional Association for Coastal Ocean Observing Systems, Maracus. In partnership with Rutgers Cooperative Extension, New Jersey Sea Grant Consortium, the Virginia Institute of Marine Science, and the Mid-Atlantic Regional Council on the Ocean. I'd also like to thank our planning team members, Susanna Music, Mike Danko, Doug Zemeckis, and Kirsten Wakefield for all of their help organizing this program. I would also like to thank Anthony Himes. Anthony is going to be our moderator this evening. We'll have 2 breaks for Q&A, so if you have any questions, please send them to Anthony in the chat, who's labeled as the moderator. For those of you who aren't familiar with MACAN, it is an association of scientists, tribal, federal, and state agency representatives, resource managers, and industry partners who seek to coordinate and guide regional observing research and modeling of ocean and coastal acidification. MACAN is co-coordinated by Maracus and the Mid-Atlantic Regional Council on the Ocean, and its efforts are focused on the waters south of Long Island to the southern shores of Virginia. The goals of MACAN are to develop a better understanding of the processes associated with estuarine, coastal, and ocean acidification, to predict the consequences for commercially and recreationally important fish and shellfish and other marine resources, and to devise local adaptation strategies that enable communities and industries like aquaculture to prepare for changing conditions. In 2019, MACAN launched a stakeholder survey to gain a better understanding of the information needs of our region. Some of you may have even participated in that survey. To give a quick overview, we received a lot of questions from the recreational fishermen about the science behind OA, the impacts it may have on recreationally important finfish and shellfish, and in what time frame we could expect to see those changes. There was also concern about whether OA impacts would be more pronounced in coastal habitats versus open ocean waters. So, in response to those questions, we've invited scientists at the forefront of acidification research in the Mid-Atlantic to discuss what ocean acidification means and what the risks are, to examine the vulnerability of coastal fishes to acidifying waters, to talk about how OA impacts shellfish and whether seagrasses can provide a local solution, and to demonstrate how 3D modeling can be used to forecast short and long-term trends in water quality and acidification in the Chesapeake Bay. Through these sessions in Hooked on OA, we hope that you'll take away a better understanding about how ocean acidification may affect the fish you target and what can be done to reduce the impacts. Before we get started, just a few navigational aids for this evening. We will be recording video and audio and questions posted in the chat box during the presentation. These recordings will be posted to the Mid-Atlantic Coastal Acidification webpage. Each presentation will be recorded and shared with the participants at the conclusion of the mini-series. Like I said before, we have over 150 people registered for tonight's session, so we ask that you please mute yourselves during the presentation so we don't have any dogs barking or dinner dishes clanging in the background. If you've logged into Zoom with an email address or a nickname, please change it to your first and last name. This will help us with our Q&A and to enter a raffle at the end of tonight's program. You can mouse over your screen name in the Zoom window and right-click on it to edit. If you're calling in by phone, you can send a private message to our moderator, Anthony, to provide your name for the raffle. If you have questions during the presentation, and we hope you'll have a lot, please type them in the chat box and direct them to the moderator. We'll take a break halfway through and again at the end to answer your questions. We will do our best to get through as many questions as we can, but if you don't get an answer tonight, please feel free to email. We'll wrap up the evening with a few closing comments, and be sure to stay on. Those participating will be put in for a random raffle for a $25 gift card to Bass Pro Shops. So be sure to stay on at the end to be able to potentially win. And now I'm pleased to introduce Dr. Emily Rivest. Dr. Emily Rivest is an assistant professor at the Virginia Institute of Marine Science, William and Mary. Her research focuses on understanding how ecologically and economically important species like oysters, hard clams, and American lobster will respond to climate change. She's an expert in ocean acidification research, conducting experiments in the laboratory to simulate future water conditions and using oceanographic instruments to characterize the dynamic coastal environments of her study species. Emily is passionate about doing science that matters, often collaborating directly with members of the aquaculture industry. She is also an avid science communicator aiming to share the importance of her work with policy, industry, and public audiences. Now I'm going to turn the program over to Emily.
Thanks so much. Good evening, everyone. I wish I could see you all, but someday soon we will see each other, I'm sure. Well, it's really great to be here. I'm so excited to talk to you about something I'm really passionate about, which is ocean acidification and shellfish. And before I get started, I'm going to get my laser pointer. There we go. So science these days is really collaborative, and although I'm the one giving this presentation today, it's really on behalf of all of the wonderful people that I get to work with at VIMS and beyond. So I want to recognize them here and our funding sources. So my co-PIs, who are the professors that I work with on the grant that funds the work I'm talking about today. And that was funded by NOAA's Ocean Acidification Program and another group within NOAA. And I'd also like to thank MACAN and Maricus and the leadership team for organizing this webinar series and inviting me, as well as Anthony for being a moderator. And then the work that I'm sharing today is from a big laboratory experiment we did a year and a half ago. And there was a whole huge team of people you can see here that contributed to that experiment. So I want to thank them too, because it would not have been possible without them. So I wanted to start off by sharing a little bit about the sort of big picture that really motivates the work that I do and how I think about ocean acidification. And for me, it starts by humans causing these really large impacts on the coastal marine environment. We're seeing global changes in temperature and oxygen levels of coastal waters, and the chemistry of the ocean is changing at rates unprecedented in the last 300 million years. And you've heard about ocean acidification before. In case you're new tonight, I wanted to go over how it works briefly, and also the parts about ocean acidification that really intersect with what I'm interested in terms of how it affects marine organisms. So we have an increase in carbon dioxide in the atmosphere caused by human activities, and the ocean does an incredible service by absorbing about 30% of that carbon dioxide. Once it enters the water, there are a series of chemical reactions that occur that cause two symptoms that affect marine organisms. So we have this increase in hydrogen ions, which means that the pH of the water goes down. So the water is becoming more acidic, more like lemon juice or soda or cola, depending on wherever you're from. And also we're seeing a decrease in carbonate ions. And this has other effects on marine organisms. So I wanted to ask folks, and I can't see the chat from how I'm sharing my screen. So maybe if someone can volunteer and read out some of the answers for me. I'd love to poll you and see from what you've learned so far in this series or what you know in general, how does ocean acidification affect marine organisms? I'm gonna try to zoom out here so I can see the chat. There we go. Don't be shy. There are no wrong answers because I really want to know what you think. Okay, softens shells, stresses them out. What else? All right, I guess everyone's really curious. Ooh, use more energy. Oh, okay, here we go. Affects reproduction, slows growth. See, I don't even need to give my talk. You already know the answers. This is great. Okay, let me get back to the presentation. Perfect. Okay, so as you mentioned, this decrease in carbonate ions affects the calcification process. It affects how shellfish make their shells, how organisms like coral build their skeletons. It makes it harder for them. It costs more energy to make those structures. And this change in pH affects a lot of the other aspects of the biology of those animals. All the chemical reactions that are happening inside bodily fluids or inside the cells are being challenged. They're being pushed and pulled, and it's harder for the animal to control those. So that's where this field of study that I specialize in, called physiological ecology, comes in. And physiological ecology can be described as the study of how an animal's bodily functions are fine-tuned to its environment. So we look at these interactions between an organism and its environment and how the two affect each other. So when I'm asking questions about how to look at how these biological responses are changing with the environment, I'm really interested in informing these larger questions of how, in response to climate change, for example, effects on the organism might scale up to influence the abundance and distribution. So where you find that oyster living along the shore, the interactions between that oyster and other species. So for example, between oysters and seagrass, or oysters and fish whose juvenile forms rely on oyster reefs, and how that scales up to affect the structure and function of these sort of complex communities that live in our coastal waters. And I do that by looking inside the animal, from the molecular level all the way over on the left here, up to the level of the whole animal, to see how it's integrating that signal from the environment. How does it tell that the pH has changed? And how does that induce a response? And then what is the nature of that response? So at the initial level of control, where different genes are being turned on and off, different enzymes, these proteins that do all the work inside your cells, which ones are more active or less active, how the animal is reallocating different energies within its budget. For example, with you in your personal financial budget, you might prioritize putting more funds into a certain area during periods of stress than you would during a time when you aren't stressed out about your finances. The same thing with an animal. We look at how it reallocates where it prioritizes putting energy. So these are the things that I think about. And so when we're putting that all into the context of climate change, what I'm interested in using this framework for is to understand how long-term environmental change will interact with the day-to-day environmental variability that already exists in our local environment. So if we think of this huge process that is ocean acidification, that's this big like ocean liner that's coming at us, it's just coming straight at us. It's going over all the little waves under it, just like ocean acidification is going over all the day-to-day fluctuations and the seasonal differences that we see in our natural environment. So when we think about how is ocean acidification going to affect oysters in the York River versus in the Potomac River, we need to really know how this really large process is going to interact with a very local process to affect what oysters are going to see in that particular place. And those oyster reefs or those oyster farms have been in place for years. And the oysters that are living there might be used to those conditions. And the oysters in the York River are used to different conditions than oysters in the Potomac River, for example. And so I'm really interested in how these local environments have shaped the biology of those animals. Are the oysters in the York River more tolerant of changing ocean acidity because of those local conditions? We'll find out. And then, completing this loop, we want to know how ultimately are these really important animals going to respond to climate change? And will those responses differ between different locations? So with that big picture in mind, what I'm going to talk with you about today is ocean acidification and shellfish and seagrasses. And this idea of whether seagrasses can help protect oysters from ocean acidification is really the overarching question or goal that's guiding this research. But my role in this project is really from the oyster perspective. So I think about how oysters are going to interact with seagrasses, but from a very oyster-centric bias. So I'm just putting that out there, that's where I'm coming from. And for all the seagrass folks in the audience, I'd love to chat with you more about your expertise and how you view this. So the way we're gonna go through the talk today, I'll first talk about global change in estuaries. I know you've heard a little bit from Grace Sava earlier in this series about ocean acidification in general, but I really want to talk more about the unique environments and what we're thinking about when we think of how seagrasses could potentially interact with oysters. And then I'll share with you some results from this big laboratory experiment we did, where we really wanted to characterize the tipping points or thresholds that eastern oysters have in our area, so we can get a better idea of where is that sweet spot in ocean acidity where seagrass can really make a difference. Okay, so first, global change in estuaries. And I'll just give you a heads up, between these two sections is when we're going to take our first break for Q&A. All right, so throughout my career, I've been involved in making ocean pH discoveries. And it's been a really exciting time to grow up as an ocean acidification scientist, because there's been so much new knowledge that has been generated in the past 10 years in this field. The figure I'm showing you here is on the, let me get my laser pointer again. On the x-axis here is just 30 days, and we have pH on the y-axis, and a high pH means non-acidic conditions, and a low pH means acidic conditions. And this is showing you what a typical 30-day period looks like in the open ocean. You can see that the pH line is very flat. There's not much change in pH going on. But with the use of the invention and the use of these sensors that allow us to measure high frequency time series of pH in the water. You can measure every hour, every minute. You don't physically have to be there to collect the sample. It's wonderful. We can learn so much about the dynamics of pH and other different ecosystems. So you don't need to look at the numbers here, just look at the shapes of the squiggles. Just by comparing the different plots, you can see that pH characteristics differ a lot between these different ecosystems. We have coral reefs, for example, that have a very regular oscillation up and down. We have other locations like along the coast of California, where we have upwelling these 2-week-long periods of low pH and really, really extreme fast changes in water pH at these other places where you have actual CO2, places where CO2 is actually venting out of the bottom of the water, of the bottom of the ocean, I should say. So this is not ocean acidification. This is just natural variation in carbonate chemistry from different drivers, not from human production of CO2 into the atmosphere. So that makes it really important to compare these different ecosystems and how they'll respond. And we've done more targeted studies, for example, where we compare different patches of habitat. So we say, okay, is one kelp forest the same as another kelp forest? Is one seagrass bed the same as another seagrass bed in terms of what do the day-to-day conditions look like? And this is an example from the coast of California where we deployed these instruments in 2 different kelp forests, which is like a very tall version of a seagrass bed. And you can see comparing the blue lines and the pink line that the variation in the pH is quite different. So if you are an abalone or a sea urchin or a rockfish living in these different patches of habitat, you are experiencing different conditions. But what's also interesting is that this distance here is, half a mile to 30 miles. So it's these distances are short enough where you could see fish movement or dispersal of early life stages between these patches. So we're zooming in on this idea that differences in the natural variability can influence marine organisms and be different in different places. So going into the Chesapeake Bay estuary, we also see these different temporal and spatial gradients come into play. And you're going to hear from Margie Friedrichs and Faye Dott in the next talk. So I'm going to give you a little tease here, and I'm not going to talk too much about it because they'll blow you away at the next one. But they produce these really great maps where they can basically predict what the pH is different parts of Chesapeake Bay. So this is a prediction of what the pH was at the bottom of the water column yesterday. And this pink triangle here is where VIMS is approximately located. And we also have direct measurements of pH. So this is a time series that was collected at VIMS, and you can see how dynamic the pH is, just like some of those other figures I showed you earlier. So one important context that I want to share is how these different time series compare and why studying ocean acidification in Chesapeake Bay is really unique and why it's so important to know what your local environment is. So this pink line that I've put on the graph here is showing you the average pH for the open ocean right now. And this red line is what our best model estimates internationally predict ocean acidification will cause the pH to decrease to in 2100 if we don't shape up our actions. So you can see that oysters in Chesapeake Bay right now, if they were living right underneath this sensor, are experiencing what we predict ocean acidification in the open ocean to be on a daily basis in June of 2019. So we need this local understanding to really be able to evaluate what these animals are used to experiencing and how they do it. It's a great opportunity to learn how do animals tolerate a pH of 7.8 if we're concerned about the pH going down across the globe to that level. So my next question for you is, oops, one moment. I'm experiencing a technical difficulty. You probably can read this, but I can't. You can read the question, so please put your answer in the chat while I solve my technical difficulty. Yeah. Okay, we got less sensitive. Oh, this is such a fascinating question. This is a question that I really want to answer. I don't know the answer to this question, but it's great to get all of your input. So I think your hypotheses are great ones that because they're already experiencing pH this low, we would expect them to, already be able to tolerate it. They're living there. That makes sense. And so what we can, sorry, I'm getting a little distracted by my technical difficulties, but what we can use this information to do, what I'm showing you on the slide, all this cool information about pH in Chesapeake Bay at these different places, we can use it to ask these kinds of questions. And this is again back to physiological ecology, is the sensitivity of these animals based on their local environment. And if we can measure their local environment, we can predict how they'll respond to ocean acidification. And if they respond to their local environment, if we can change their local environment by, for example, restoring seagrass in that area, then we can help protect them from experiencing ocean acidification. So the other important points I want to make about this is that if we can use this figure of these spatial differences, we see these large gradients and change in pH throughout Chesapeake Bay and throughout a lot of oyster habitat. We can see that it changes by 0.4 to 0.5 units across this space, which is greater than the amount of global ocean acidification by the end of the century. And oysters can also experience that same magnitude of change over just 24 hours. So we have so much to learn by just looking at how these animals can perform well in their natural environment. In addition to this global driver of ocean acidification, we also have other local drivers. So we have those drivers that are causing that huge day-to-day cycling of pH in the water and other kinds of changes in pH. And so Grace may have shared this figure with you already, but we have things like freshwater input, we have runoff, so pollution or nutrients that cause eutrophication, and then this nutrient pollution can cause these algal blooms to form, and when those blooms die, they get digested by all the bacteria in the water, and that produces a ton of CO2, which causes the pH to go down, and you get a local acidification event. So there are lots of different ways that oysters can experience acidification. Another really cool thing that causes the chemistry of the water in Chesapeake Bay to be different is the river water. And there's some new work coming out of Penn State, and Margie is also, and other folks at VIMS are collaborators on this work, that's looking at a different measure of chemistry of the water called alkalinity. And alkalinity refers to the buffering capacity of the water, so how easy it is to change the chemistry of the water. And these colored lines here correspond to data coming from each of these tributaries feeding into Chesapeake Bay. And you can see that each of these lines falls at a different height along this chart. And what that means is that each of these rivers has a different buffering capacity. So if you're an oyster in the Potomac River here, which has a higher buffering capacity, you'd be more protected from experiencing rapid change in the chemistry because the water is naturally a better buffer. But one of the other things is that if we have increased stream flow, so that's increasing to the right here, you see that the buffering capacity goes down. And one of the predictions for our region is that with climate change, we're going to get more rain in the spring, in the winter and spring. And when that happens, we'll get more stream flow. And so overall, we can predict that our water will have less buffering capacity. So a wetter future means a weaker ocean. And that's because of the way that this buffering capacity works. So here is a zoom into those squiggles that you were looking at on some of the previous figures, where we see that pH is going up during the day and it's going down during the night. As ocean acidification progresses, we're going to see this a decrease in the average pH, what you're looking at here in this red line. So this is the average pH for each of these squiggles from pre-industrial to 2015 to 2100. But what we're also going to see is, because of this wetter future, a decrease in the buffering capacity, that these daily swings in pH are going to become larger. So in a coastal environment, it's not just that ocean acidification is causing things to become more acidic in multiple ways. It's not just the average state that's becoming, the average water condition that's becoming more acidic. You're seeing that the daily change in pH is becoming bigger as time goes on. And so the difference between the highest pH in the day and the lowest pH at night is becoming bigger. And this aspect of change, the animals, if their physiology, if their biology is really tied closely to the water conditions, they'll have to do a lot more work to figure out what to do as the pH is changing so much. But what this also means is that animals and plants in the environment are in the surrounding habitat are playing a larger role in affecting their environment. That's another way to think about it. Because if you think about what's causing the pH to go up and down, it's the photosynthesis of the seagrass during the day that's removing CO2 from the water and therefore causing the pH to go up. And then when the sun sets and you don't have photosynthesis happening anymore, it's all the respiration, all the CO2, all of the living things exhaling CO2 in the water that's causing the pH to go down. And so really, when thinking about ocean acidification, studying the biology becomes even more important when we think about how this global process is progressing, because all the plants and animals in the habitat are going to be playing a much larger role in the day-to-day experience that's bathing everyone. That makes sense. So that's where seagrasses come into this story when thinking about oysters. That seagrasses are one of those important components of the local ecosystem that shapes the surrounding habitat. And here what I'm talking, they shape the surrounding habitat in a lot of ways, providing structure, providing food, providing hiding places, providing nursery habitat, providing all kinds of ecosystem services. But what the service that we're really interested in with this project is the way they're affecting the chemistry of the surrounding water, because that's what the oysters would experience. And so again, they do that during the day through photosynthesis that's causing the pH to go up, and during the nighttime with respiration causing the pH to go down. And so understanding how seagrass will respond to ocean acidification, how they respond will affect what all of the critters that live in and among the bed. And also, that water is moving through the bed and traveling someplace else. So there's probably some zone of influence around the bed that experiences what the seagrass do to the water. Okay, so to wrap up some takeaways from this first part of the talk, the chemistry of water in Chesapeake Bay, where we're studying oysters, is really variable. We have daily ups and downs, and we have differences in the chemistry of the water among locations. It's important to know that there are local factors like runoff that can intensify acidification. And this is also a good takeaway to think about in terms of what you can do to help with respect to climate change and ocean acidification. Sometimes those local factors are much easier to tackle at the individual or community level. And then this other takeaway is that biological processes will also play a really important role in influencing what organisms like oysters or seagrass are going to experience in the future. So ocean acidification isn't going to happen the same way everywhere. And it's really important to understand context. So in other words, time and place matter. And that's why as a biologist, I have spent a lot of time learning how to use oceanographic sensors to characterize what exactly these oysters are experiencing, because it's often very different from what the NOAA buoy at the mouth of the bay is measuring, for example. And with all of that being said, what leads into the next part of the talk, what we've learned about these different times and places and how chemistry varies throughout, that oysters in Chesapeake Bay likely have different OA sensitivities than oysters elsewhere. So with that, I'd like to pause at this point in the talk and take any questions you might have so far.
Great, thanks, Emily. So I see Kirsten dropped a message in the chat, but anyone, if you have any questions, feel free to send them in now. We do have one question so far from Edward, and he asked, does increased acidification impact the fertilization rate or effectiveness of shellfish or other broadcast spawners?
Yes, it does. I'm not aware, and Anthony, you're welcome to chime in too, since I know you are an ocean acidification scientist as well. I'm not aware of any studies off the top of my head that have looked at that in oysters, but I haven't read up carefully on that. But I do know that in sea urchins, there's been a lot of work looking at fertilization dynamics and looking at sperm motility, for example, that it is negatively impacted. There's also the pH plays a really important role in controlling a lot of biological processes. And so, for example, when fertilization first happens, when the egg first gets fertilized, there's an envelope that has to pop up that prevents other sperm from fertilizing that egg. And at some extreme pHs, that process is disrupted. And so you can get polyspermy, and then you won't get a normal development of that embryo, at least in sea urchins. Good question.
Okay, I don't see any other questions right now, so you can continue on as you like.
Thanks, Anthony. And thanks everyone for participating so far. It's fun. I miss getting to interact with you otherwise, so. And the technical difficulties are me just trying to be able to see the chat box while I'm sharing my screen. All right, so next we're going to move into the second part of the talk where I'll share some preliminary results from this big laboratory experiment we did in the fall of 2019, where we characterized the responses of juvenile and adult oysters to ocean acidification. And this is the project that was funded by NOAA's Ocean Acidification Program and NCCOS, which is another coastal ocean program within NOAA. And this program was really focused on looking at ocean acidification thresholds. And this figure here is a nice one to show you the idea of a threshold. Here on the x-axis, we have a parameter of acidification. So going to the left, acidification is increasing. So near we have 0 and 1, it's very acidic. And on the y-axis here, we have the proportion of normal development in oyster larvae and mussel larvae. So a 1 here means that 100% of them developed normally. And so if we start over here at no ocean acidification conditions, you can see we eventually reach a point where we start to have this precipitous decline in the percent of embryos that develop normally. And so these are the kind of thresholds with oysters that we're interested in detecting. Conditions of water chemistry beyond which they can't survive, grow, or reproduce, or we can insert here any other process about them, like calcification, for example, that we're interested in. So when you look at these graphs and think about ocean acidification as time going from present day to the future, which one of these will pass their threshold first? Based on where the orange lines are. There are no wrong answers. What do you think? Okay, we've got a vote for oysters. Mussels. Right. Based on these graphs, I think that the mussels will pass their threshold first, because they have a threshold at a higher, closer to present day conditions. It's a 2.75 or something like that, whereas this is more like 2.3. So as ocean acidification is progressing, it'll hit this 2.75 threshold first. So when we're comparing, instead of mussels and oysters, we're comparing 2-year-old oysters versus 6-month-old oysters. We're doing these kinds of comparisons to see which one is more sensitive. So we'll get a chance to revisit this question in a bit, looking at our oyster data. So great job, everyone. Thank you. So I may speak for everyone to say we don't want the oysters to reach those tipping points. And so how can we mitigate that? We need to understand where that tipping point is, but what else can we think about that might be at our disposal? Why would we want to do this, and how would we approach it? There are a couple of contexts in Chesapeake Bay where humans are interacting with oysters, and one is through aquaculture, and the other one is through restoration. And through both of these, in addition to the very important economic role that aquaculture provides, these oysters in the bay are also, while they're in the water, providing ecosystem services like taking up nutrients and improving water clarity, and providing habitat for the fish you like to catch, for example. So we want to prevent them from reaching these thresholds. We also want the big investment, monetary investment that, for example, all the states in the Chesapeake region, the federal government investing in restoration of 10 bay tributaries by 2025. We want that to be successful. We want all those tax dollars invested in restoration to be a good investment. And so, understanding when oysters will reach these thresholds and how we can avoid them is very relevant in that context. In the same way with oyster farming, if we can understand where oysters will reach these thresholds first, that can help us guide farmers for, leases that will be particularly good, or, where seagrass is needed to protect a particular part of the lease, that kind of thing. So with this particular project, we're looking at whether seagrasses could be a solution in terms of helping oysters avoid experiencing those thresholds. And as I mentioned in the first part of the talk, seagrasses could do this by the way that they change the chemistry of the surrounding water. The fact that they remove CO2 during the day and cause the pH to go up might cause the pH to go above the threshold, or might mean that the average pH throughout the day is above the threshold. So we know that SAV can modify the carbonate chemistry, but the missing link for me personally was, is it enough? Do they modify the chemistry enough that we will actually see a measurable benefit to the oysters? And this was a really critical knowledge gap for me, for us to fill when thinking about, could this be a practical solution? And thinking about this in the Chesapeake Bay region, we really needed to know, for the oysters that live here, where are their tipping points, so that we can combine that with our knowledge of what seagrass beds can do to modify the chemistry, to be able to say, okay, do those two things fit together well? Would this actually work? And it's really hard to do that by doing an actual experiment or going out into the bay and trying to measure these things directly. And so this team of people that we have together, we're embarking on this modeling effort to try to answer this question. So in this modeling effort, we use mathematical equations to represent all those processes. And we can run different simulations with different water conditions, different characteristics of the seagrass bed. We can put oysters, with at different heights within the bed. We can change the size of the bed. There are all of these different kinds of scenarios that we can run to try to find that magical combination of what would work the best in terms of protecting oysters. And so where we started was with this eco oyster model, and it has all these cool inputs that go into it. It has phytoplankton, it has aspects of the sediment, it has nutrients in the water column, and it simulates water going to and fro in the model, and it can tell you how the oyster will respond. But what it doesn't have is aspects of carbonate chemistry. So it could tell you how changes in temperature or oxygen or nutrients affect the oysters, but not carbonate chemistry. So through this project, we wanted to develop those mathematical equations to put into this model that would allow us to predict, using the model, how ocean acidification will affect the oysters. So this is sort of generally what the model looks like without ocean acidification. We have, again, these are representative of the equations that we have the tissue weight of the individual that can increase because it's eating by filtering the water. It's respiring, it's creating waste, it's building its shell. All these arrows refer to different things that the model is calculating. So what we want to do is we want to add ocean acidification into this. So here where this represents adding all these functions based on the pH of the water, how that's going to affect filtration, how pH will affect respiration, how pH will affect the generation of waste, how it will affect the formation of the shell and the dissolution of the shell. And in order to do that, we needed to run an experiment to generate data. And then using the data, we could come up with all these mathematical equations that we could put into the model. And so ultimately, we wanted to get at this question of what ocean acidification thresholds exist for eastern oysters in Chesapeake Bay. And this was the general question guiding our experiment that would generate the data to make all these formulations. So for this experiment, we took oysters of two different ages. We had oysters that were 2 years old, and we had oysters that were 6 months old. And we had them in the laboratory where we controlled the chemistry conditions, and we had 4 different scenarios of water chemistry that we used. And so these oysters were kept in the experiment for 2 months. And this is the facility at VIMS. That's part of my lab group where we ran this experiment. So we had oysters in a bunch of these different tanks for 2 months where we were taking care of them. And at the end, we measured a variety of different biological processes of the oysters. So everything from survival to condition index, which is a metric of just general oyster health. We measured calcification, we measured respiration, which is an estimate of how much their total energy needs at that time. We measured how much waste they were producing. We measured how quickly they were feeding. And so I'll share some of these results with you today. Okay, so the first thing that I'll share with you is condition index. And I want to walk you through this graph because I like showing graphs and I want to make sure you all can understand it. So what we have here on the x-axis is, again, another metric of ocean acidification, this time pH. And on the right side, again, we have scenario of no ocean acidification. And on the left side, we have very acidic conditions, respectively. And then on the y-axis here, we have condition index. And condition index is the percent of the total mass that's made up of tissue. So a high condition index would be where you have traditionally interpreted as a healthy oyster because there's much more tissue with respect to the total weight. And that reflects a healthy oyster because oysters that have a lot of tissue usually have a lot of energy reserves. They just have a lot of glycogen, a lot of fat that's stored in their tissue. So when we see a high condition index, we typically interpret that to be a healthy oyster. But we're not seeing that here. So what you can see from this graph is that these lines indicate what the model formula that we can make from these data. The red line is for the half-year-old oysters, and the blue line is for the 2-year-old oysters, and the dots are the actual data points that we measured. So if you just want the overview, look at the line and the direction it's going in. And so what we can see is that as pH decreases, as ocean acidification becomes more intense, we're seeing that condition index is actually increasing. What? That doesn't make sense to me based on my hypothesis. Based on what I've shared with you so far, we could say, oh, well, are oysters actually going to be healthier in the future? They have more energy reserves? We were very puzzled by this. And so we did some follow-up tests to see what could be going on. And here's an example of what we would expect with condition index. So why does condition increase with ocean acidification? Here is a picture of, these are actually not Crustacea virginica, they're different species of oyster, but a good comparison for what different condition indexes typically look like. Where you have this oyster here on the left that has a high condition index, you can see there's much more tissue filling out the shell. The mantle is much, this tissue here is much less transparent. Whereas this oyster, if you shucked that oyster and opened it, you would be very disappointed thinking about eating that. And you would have spent a lot of energy shucking that oyster for the meat you got out of it. So, when we look at these data, is it really the tissue that's bulking up that is generating this high condition index, or is there something going on with the shell? So based on how we calculate this, with this total mass in the denominator, if the total mass includes the weight of the shell, if the shell weight is going down, that could cause us to calculate a higher condition index. So we needed to figure out what was going on with the shell. And this brought up a really important point that the way we think about condition index may not be reliable in the future under ocean acidification if we're thinking of it always as only the tissue mass that's changing, that's causing changes in condition. So we need to be tracking shell condition in order to really understand how to interpret condition index when we measure it. So I will leave you in suspense about what's going on with the shell because it will come up in a minute. So in addition to measuring oyster condition, we measured a variety of other things. We measured respiration, calcification, and excretion using this really cool setup where we put oysters in sealed jars and we measured how the water changed around them over a couple of hours. So using these probes, we could measure how the oxygen concentration in the water was going down as the oysters were breathing. We could measure how the chemistry of the water sealed in this jar was changing as the oysters were calcifying and removing those carbonate ions from the water. And we could also sample this water in the jar at the end to look at all the waste that the oysters were producing during that time. So I'm going to share with you next the results for calcification. So calcification refers to the formation of the shell. And if you see a positive value here, so anything above the x-axis indicates shell formation. So the shell is growing, it's increasing in weight over time. And everything below the x-axis here is actually shell dissolution. So even though the animal is still alive and is still potentially laying down, forming new shell, overall the shell is dissolving and the weight of the shell is going down over time. And so what you can see is that what our lines here are showing us is that as ocean acidification progresses, we are going from a current ocean where we have oysters that are increasing the weight of their shell over time to a future ocean where the weight of an oyster shell is decreasing over time. And this is you might have oysters who aren't able to make a shell essentially in this. We started off with oysters that already had shells, but if you think about an oyster completing its life cycle in its environment, it might be hard for oysters to have a shell. This is not at a close time point in the future, so don't get too upset. But it does, provide a really important context about these thresholds that we're trying to avoid, right? So one thing about this figure is that there weren't any differences in between our two age classes in this figure. When we were looking at the live animal and how its calcification is changing over time. We wanted to learn more about what was happening to the shell because of this puzzle with our condition index. And so when we were done taking these measurements, we took those oysters, we shucked them, we dried the tissue to take other measurements, and we put the shell back in the chamber without the animal associated with it anymore. So it's just the shell. And we repeated our measurement to see what the shell was doing during this time. Because ultimately, when the animal is alive, this trend that you see here represents what the animal itself is actively doing and what the water is doing to the shell that's outside of the animal's control. So we wanted to be able to separate those two parts and say, okay, what is the water doing and what is the animal doing? So by looking at the shell by itself, we can get at what the water is doing to the shell. Okay, so you have shell forming, shell dissolving. Before I move on to the next part, I wanted to show you that this zone here, so this, where it's crossing the x-axis here, is where we could interpret a threshold to be, where that transition from the shell forming to the shell dissolving is right in this sort of 7.6, where's my mouse, 7.6 to 7.4 pH area. And I just wanted to put that into perspective for you with the figure I showed you earlier of the time series of data from June in 2019. This is a zone that oysters sometimes see at night, where they might get into a situation where they're net dissolving for a few hours. So it's kind of, it's an interesting, it's good to keep that context in mind. Okay, so this is what the data look like when we're only looking at the shell. So we expect to see more dissolving because the animal isn't there to make any new shell. So that's not a surprise. But what I want you to notice is, unlike the last figure I showed you where the young oysters and the old oysters were responding in the same way, when we just have their shells, the shells are doing different things. The shells dissolve at different rates. So here, let's take this blue line first. This blue line is much steeper than the red line. This blue line is for our 2-year-old oysters. Okay, and so you can see here, this number is where that, that line crosses the x-axis at pH 7.71. Okay, now compare that to where the red line crosses the x-axis. This is for the half-year-old oysters. It crosses the x-axis at a lower pH of 7.47. So my next question for you, this is the follow-up question to the oysters versus mussels question. Which age oyster is, in terms of their shell, is more sensitive to ocean acidification? Which threshold gets passed first? Okay, we've got Faye, Carl, Rich. Oysters, yeah. Anyone else? Okay, it's the 2-year-old oysters that their shells start dissolving at a higher pH. So in terms of, years into the future, they're going to start, yeah, that's exactly right, Michael. They will start not dissolving sooner than the young oysters will. So if you think about, okay, this is the same animal, right? They're just a year different in age. They make their shell presumably the same way. So why would the shells be dissolving at different rates? Two-year-old shells dissolve faster. One second. Okay. Why do the older oyster shells dissolve faster? This is something cool that could be an application to aquaculture, I think, which will be an interesting direction of this work. This is just my hypothesis, so don't hold me to this. And if you have any other insight, I'd really love to know. But the shells are made out of the same material. But one thing we noticed, and this is why it's so important to observe lots of things while you're doing your work, that the older oyster shells, they're riddled with all these holes and nooks and crannies that come from parasites from these other critters that are living in and on their shell. For example, these worms will create burrows in their shell and live in their shell, the mud blister worms. I'm sure if you've eaten oysters, you might have seen this discoloration inside the shell. Those are from the burrows of the mud blister worms. But what is happening is that by creating those burrows, these critters are increasing the amount of surface of the shell that's exposed to the surrounding water. And with the young oysters that we were working with, they were very smooth. They hadn't been colonized yet by all these different parasites and critters that could make these burrows or make little homes in the shell of the oyster. And so if you were to take two shells of the same size, one that was smooth that didn't have any parasites and one that had all these burrows in it, you would expect the one with all the burrows to dissolve faster just because there was more of it that's exposed to the water that could be dissolved. So that was just a really neat surprise that came out of this work. But the oysters need their shell. The shells of the oysters are really important for all the services that oysters provide. And so, if we can prevent the shells from dissolving as quickly, then we might be able to prevent those services from going away as quickly. So putting the half shells together, we learned that combining all these data together about, how the shell is changing and how the tissue is changing, we've learned that this change in oyster condition that we were measuring was really caused by shell dissolution, that because the shell was dissolving, it was making it seem like the oyster's condition was improving over time at ocean acidification scenarios, which wasn't true based on how what condition index traditionally means. With calcification, we saw different responses are different mechanisms that are really affecting the oysters at different ages. So with these 2-year-old oysters, their calcification, this decrease in calcification as ocean acidification progresses, is really driven by this uncontrolled shell dissolution. And so an intervention that we could have perhaps through seagrasses is to improve the surrounding water chemistry, and then the shell won't dissolve as quickly or at all. But with the half-year-old oysters, that net calcification is not, their shells were much less sensitive to dissolution. And through other measurements that we've made, it's really that change in calcification over time, that decrease, is really controlled by the animal itself, trying to make changes to stay alive under those more acidic conditions. And so the intervention there to help the really young oysters might be to prevent, in addition to removing ocean acidification in general, would be to avoid the presence of multi-stressors. So I wanted to share a little bit about some of the other things that we measured. We also measured filtration rate. So we put oysters in jars and we added some food, some phytoplankton to those jars. That's what causes just like when you see a phytoplankton bloom in the bay, that's what causes the water to look green and kind of murky. And we measured how much they ate over time. And we didn't find an effective pH, but one of the things that we did find was that the oysters didn't eat very consistently. And so that's something that we are putting into the model. You can see the percentages here where the percent of the oysters that actually ate during our experiment. Maybe they had performance anxiety, you know, they were in the jars and they didn't like the jars, and but they didn't like the jars any better at high pH than they did at low pH. But we're looking into this more because in the models that we're using to predict this, they assume that oysters are eating all the time. And it's clear that they're not. So this is a really important thing we need to take into account when we're trying to predict how they will respond to future ocean acidification. So to wrap up some insights on our ocean acidification thresholds, that condition index as we've traditionally measured it may be unreliable in the future without also tracking shell condition. So that's an important thing to think about. That adult oysters were more sensitive to ocean acidification than juvenile oysters by up to 0.3 pH units, which is a really large change in pH. And now that we've completed this experiment, we have this cool data set for developing those formulations. So we're putting those formulations into the model I showed you towards the beginning so that we can run different scenarios. And so what we're going to do now that we have those ocean acidification functions in our oyster model, we can run these different future scenarios to look at the effects of ocean acidification on oysters. So we can include seagrass in that model, and again have the pH go up during the day and down at night, more so because of the seagrass, and see if that changes the way that oysters respond to ocean acidification. And we can also use the model to evaluate how the benefit of the seagrass to the oysters changes depending on the characteristics of the seagrass bed. So for example, how deep the water is or how big the meadow is. And it's so easy with a model to be able to try out all these different combinations. So to bring it back to seagrasses in the end, I want to put this back into an environmental context. And these are two data sets that were collected by, and I didn't put this on the slide, but I'll say it for you and for the video, by Ken Moore and the Chesapeake Bay National Estuarine Research Reserve Program at BIMS. And you can see these squiggles again, right? We have 3 years of data for each of these plots. We have 2 different coastal bays on the eastern shore of Virginia. And so look at the squiggles, and I'd like you to weigh in which bay do you think has seagrasses? Okay, we have 1 vote for South Bay, spider crab. South Bay. Great. Well, the answer is that they both do. So you're all right. But South Bay has much more abundant seagrass, much more dense bed, and Spider Crab Bay is in a recovering seagrass situation. So you can see here in this left-hand panel that there's much more time where the water is at a much higher pH, and that could likely be due to the very vibrant seagrass beds that are in these coastal bays. So where do you think oysters will do better? Got some votes for South Bay. Depends on their age. Yes, that's great. Yeah, we are going to be running these models to try to figure this out, and we're using these data sets from the coastal environment, and we're using what we know about these seagrass beds from our colleagues who work there to be able to design these scenarios in a very relevant way to our local ecosystem. And I've thrown up in case you're interested, these red and blue lines that represent those tolerance thresholds. So the 2-year-old oyster threshold is right here, then the red line, and the blue one is the 6-year-old threshold. Okay, so we haven't talked a lot about recreational fishing, but certainly seagrasses and oyster reefs are important important habitat for a lot of fish species, commercial and recreational species. And so based on what you've learned today about how ocean acidification is affecting oysters, what do you think that might mean for the fish that live there or that rely on them for at least, part of their life? We have a wide diversity of responses. This habitat is so this habitat is so important, and it provides so many different kinds of services, just from the structure that's there, those hiding places, to the chemistry of the water. And so it's a really complex question to think about how these broader changes are going to affect habitat for fish. Fish have the very convenient ability to move away that oysters do not, if they don't like the water conditions in a particular place. So maybe they will spend more time in seagrass beds where the pH is higher in the future. We will need to do more research to figure that out. But I wanted to thank you so much for all your participation tonight. It's really hard to give a talk to your computer and not see the people that you're talking to. And I've really enjoyed being able to connect with you through the chat, and I hope we have a chance to meet and interact more in the future. To wrap up, I hope that some of the things you've taken away today are that global ocean acidification will translate to different local threats that will affect seagrass and oysters, and that adult oysters are more sensitive to ocean acidification, at least in terms of their calcification, and will likely benefit more from seagrass effects. And with that, I would love to take any questions that you have.
Great. Thank you very much, Emily. We do have some questions rolling in, but if anybody still has more, please type them in the chat box. So our first question is from Kathleen, and she says she lives near Delaware Bay and is interested about how your results may apply to Delaware Bay, and if you know if there are similar data sets for oysters within Delaware Bay or any collaborations with the Jacques Cousteau near that manages Delaware Bay.
Is that the National Estuary Research Reserve? Is that what you mean?
Yes.
Okay, great. Well, certainly that National Estuary Research Reserve, I don't have any direct collaborations with them, but I do know that there is an ocean acidification working group among a lot of, among all the different NEAR programs around the US. And so I've interacted with that program in that way. And they're also part of our MACAN group. So we do talk a lot about how to measure ocean acidification in these coastal environments. We talk about different instruments and way to do it, and how we can do it in the same way so that we can compare our data and do these kind of experiments where we're testing the sensitivity of oysters in Chesapeake Bay versus Delaware Bay, for example. I don't know of any similar studies going on with oysters in Delaware Bay at this time, but certainly a lot of interest in ocean acidification in Delaware Bay. And there's so much research going on with oysters that it's entirely possible that I'm just not aware. Great question.
Okay, so our next question is from Claudia. They say they've heard of seagrass or kelp plugs being used in the open ocean as CO2 sinks. After a period of time, they get so large that they sink and effectively remove carbon from the atmosphere. Do you know anything about these systems or how they function?
Yeah, there has been a lot of interest in underwater plants and algae like seagrass and kelp, and how they can be used with respect to ocean acidification and climate change. And so in order for them to serve as an actual sink of carbon, for them to remove carbon from the water, from the atmosphere for a long enough time that will make a difference with ocean acidification, it needs to either be buried in the sediment or sink to the deep ocean. And that's really hard to do. There's been a lot of work looking at seagrass beds, actually, because when the plants die off in the wintertime and the blades break off at the ends, that carbon that was taken out of the water and turned into plant material can then either be digested and released back into the water as CO2, or it could be buried. And so other folks have looked at, how can we manage seagrass beds so that there's more burial and therefore more of a sink. But most of the work that I'm familiar with like suspending those plants in the open ocean has been more around farming and using them as a kind of greenhouse, for example, to co-locate them with shellfish. And it's just not for the sink aspect, but just for the instantaneous way that they're improving the chemistry of the water that makes it better for the oysters. Lots of different ways they can help.
Okay, so our next question was in reference to the two different year classes of oysters you were showing. And Kathleen asks if the two different year classes had similar shell characteristics such as depth or size or shell density, and if the half-year-old oysters and the two-year-old oysters came from the same location within Chesapeake Bay.
Yeah, those are really great questions. I haven't calculated density, but I could. So that's a great thing, next thing for me to do. That's a great suggestion. They were definitely different sizes and different weights. I can go back to that. It'll take me a while, but I will get there. That photo overhead shot I showed, oh, there we go. They're definitely different sizes, but that's, one nice thing about condition index is that you would expect the shell and the tissue to grow in kind of a similar way as the oyster ages. So we would expect that that measurement would have accounted for that if they were in the same treatment. These oysters we got from our research hatchery at Vins, and since they're different ages, they have experienced different water conditions during their lifetimes because one has only been alive for 6 months and one has been alive for 2 years. So we can't really avoid that aspect. But they were held under the same conditions before we started the experiment. And they're from the same family of oysters that our hatchery maintains. So as similar as we could get.
Our next question is from Carl, and they ask, what organisms do you think will become relatively more abundant in environments like the Chesapeake Bay under more acidic conditions?
Well, jellyfish is my answer. There's been a lot of speculation as well over whether photosynthesizers like seagrass will become more abundant. But as you all likely know, seagrass, for example, has been limited in its abundance historically because of eutrophication. So just because ocean acidification is happening and providing more carbon for them to use to create their tissue doesn't necessarily mean that they'll be able to benefit from it if those other stressors exist. So it is a bit hard to predict when there are so many different drivers that affect it. But certainly one of the predictions with ocean acidification is that these calcifiers are going to have a much harder time. So the squishy things will do a lot better. But we'll figure out a way. We won't get there.
So our next question is from Rich, who says they grow oysters in a surface float in a creek off the Potomac River, and they ask if the lack of proximity of seagrass habitat affect oyster health.
So because they don't have seagrass close by, they're wondering if that affects their oysters' health?
Yes.
Okay. I mean, the oysters are, at least right now, I don't know exactly where you are, what your water conditions are, but they have the ability to be healthy in water conditions right now. So I would say that it isn't necessarily a bad thing that the seagrass aren't close by. And, one of the things that we're thinking about with this project and thinking about how oysters and seagrass could interact is right now, at least in the state of Virginia, our regulatory framework through the Virginia Marine Resource Commission prevents you from disturbing any bottom that seagrass move into. So in terms of trying to intentionally place oysters near seagrass is not allowed necessarily right now. But so we're hoping that this kind of research can help inform new policy. If we can show that there are benefits, that there are measurable benefits and valuable benefits in terms of potential economic benefits, cultural and ecological benefits, that maybe we can change those regulations to allow for these kind of beneficial interactions. But yeah, I think your oysters are okay where they are.
Great. Looks like we have one more question here from Kathleen. They ask, they mentioned that scientists in Delaware Bay see some resistance with fishermen on being careful around seagrass beds. They ask if there's a similar problem in Chesapeake Bay and if there are efforts to restore seagrass beds in Chesapeake Bay.
Mm-hmm. Yes, there's a long and very dedicated history of trying to restore seagrass in Chesapeake Bay. And I'm not as familiar with interactions between fisher folk in Chesapeake Bay and seagrass beds in terms of, going where they're technically not allowed to go or damaging habitat and things like that. But I think, this question really highlights all the different priorities and needs that we need to weigh in terms of thinking about how humans interact with the coastal environment, and that the science may suggest that we need to leave seagrass alone and not run boats through it and things like that. But also, humans are gaining a lot of value from the ecosystem, and so these are things we just need to take into consideration moving forward. I'll be interested. That's a really good thing to keep in mind for us when we're thinking about, going out into South Bay and Spider Crab Bay to study the seagrass beds there through our model over the next year in terms of how folks interact with those beds. Thank you for that question.
All right, so that is the last question I see. So with that, I'll turn it back over to you, Kirsten, to wrap us up.
Thanks, Anthony. I'm going to go ahead and switch us over to the raffle for